In this new episode, we are going to challenge one of the biggest conceptual mistakes in agriculture worldwide, the idea that soil pH blocks the availability of certain nutrients for plants and creates nutritional problems in crops. For the purpose of this video, let's assume we all have a basic understanding of chemistry and that we know what we mean when we talk about pH or hydrogen potential. This chart is very well known in agronomy. It has appeared in books, courses, and presentations for decades. So, what is this chart trying to show? The idea behind the graph is simple. The horizontal axis represents soil pH from acidic to alkaline. Each bar represents a nutrient, and the wider the bar, the more available that nutrient is supposed to be for plants within that pH range. Basically, this graph suggests that there are pH ranges where nutrients are available and other ranges where they are not. This idea makes sense and can even be considered true in quotation marks if we look at agriculture from a conventional point of view. In that model, soil is seen as an inert substrate, a physical support for plants, and a chemical storage of nutrients. But reality is far more complex. The interactions happening in soil are so numerous and interconnected that is almost impossible to simplify them for easy understanding. And despite all the scientific advances of the recent centuries, we still do not fully understand most of the processes and interactions that occur in the soil. Many other factors are involved, such as biology, organic matter, clay type, root activity, and many more. For example, today we know that plant roots can modify pH at microscale around them. We also know that microorganisms can make nutrients available even at pH levels far outside the classic range of 6 to 7.5. Because of all of this, pH is not the determining factor for nutrient availability. A soil can have an ideal pH and still be biologically dead. At the same time, a soil can be acidic and function perfectly well, as we see in forests or natural grasslands. This chart may work very well in a laboratory, but it does not represent what happens in real soils. Let's look at a simple example. We have two soils with the same pH of, let's say, 5.2. One is compacted, lacks structure, and has poor root development. The other one is spongy, dark, and has deep roots. Chemically, they have the same pH, but biologically, they are completely different worlds. So yes, the pH chart is not lying, but it does not tell the whole story. Another idea that comes from this graph is the simplified belief that when soil pH decreases, aluminium becomes soluble, toxic, and something that needs to be neutralized. Well, actually, soil aluminium has two sides: integrated soils with low biological activity and little organic matter. Aluminium tends to be in a free form, meaning it's present as aluminium 3+. In this form, aluminium can negatively affect root growth and overall soil life. The opposite situation occurs in living, balanced soils with sufficient organic matter. In these soils, aluminium binds strongly to humic acids, fulvic acids, and phenolic compounds of plant origin. This creates very stable organomineral complexes. As a result, microorganisms cannot easily break down the carbon in these compounds, which reduces carbon losses from the soil. That is why many soils rich in aluminum and iron can store carbon for decades or even centuries, including warm and humid climates. When aluminum is part of these stable organomineral complexes, it acts as a bridge between particles. As a highly effective trivalent cation, it helps bind clay to organic matter, humus to iron oxides, and microaggregates to each other. Aluminum also interacts with iron, forming oxides and hydroxides that act as a natural cement in the soil. These compounds stabilize microaggregates and reduce clay dispersion. These bonds are stronger and more durable than those formed by calcium alone. And all of this is particularly true in tropical soils, volcanic soils, and highly weathered soils. Without aluminum and iron, many of these soils would collapse structurally. The behavior of an element depends on soil type, mineralogy, biological activity, and management history. Though aluminum cannot be judged in absolute terms, it is not always the villain. Nature did not make a mistake by putting aluminum in the soil. Like everything else in nature, aluminum has a function when the system is in a dynamic balance. Our real challenge in agriculture is not to fight natural processes, but to understand them and learn how to work with them. Regeneration begins exactly there, by stopping the imposition of general rules and starting to read the soil in front of us. What we see in this image is a tropical soil in Colombia, where organic avocados are grown in a soil with a pH between 4.6 and 4.9. This is a very acidic soil. However, despite this low pH, the crop is in excellent health condition. Root development is strong and soil aggregates are clearly visible. If we think about phosphorus, according to the pH nutrient availability chart, this element should not be available in such acidic conditions, and yet these plants are healthy and productive. The same thing happens in tropical forests with acidic soils, where we see an enormous diversity of plant life. So what does this tell us? It tells us that plants are accessing phosphorus. That means there is biological activity and overall soil health that allows phosphorus to become available. If we look at soils in natural ecosystems around the world, we find plants growing across all pH values. And this is very important to understand because otherwise plants would only exist between pH 6 and 7.5. The only places without plants are those limited by other factors, such as lack of water, for example. Now, the question could be, how do we correct soil pH? Well, we do not need to correct anything. There is nothing to correct in a soil with a naturally defined pH. Nature did not make a mistake. What we need to do as humans is adapt and stop going against natural systems. If soil pH has changed over time due to intensive agrochemical use, then the solution is not to adjust pH so we can continue the same practices. The solution is to change those practices and implement regenerative ones that allow the system to recover its health. We must regenerate ourselves at the same time we regenerate the soil. And by regeneration, I mean changing our mindset, our paradigm, and how we perceive the world around us. Let's imagine this situation. We arrive at a soil that has developed balance and biodiversity over millions of years. We remove all that life and replace it with a crop chosen only for profit. A crop not adapted to that soil or its pH. When problems appear, we blame the soil and we say the soil needs to be fixed. But is it really the soil, or is it our way of thinking? Agricultural lime is often presented as a solution to fix the pH, but it only fixes the problem in the short term while creating bigger problems later. The widespread use of lime to correct soil pH has negative effects on soil health. Rapid pH changes disrupt microbial communities adapted to that environment. These also accelerate organic matter oxidation, increase carbon loss as a carbon dioxide, and weaken soil structure. Relying on lime reduces system resilience, increases dependence on external inputs, and moves soils further away from biological self-regulation. The next image shows a research conducted in Argentina in a well-known wine-producing region called Mendoza. Soil samples from both situations were collected and used to produce soil chromatographies. For those who are not familiar with it, soil chromatography is a visual diagnostic technique developed in the first half of the 20th century. Soil chromatography evaluates soil quality through patterns formed on filter paper. Through a reaction between soil and extract solution and a reagent, a circular image is produced. The colors, shapes, and transitions reflect how the soil functions as a living system. Instead of giving us only numbers, chromatography shows biological activity, organic matter quality, balance between mineral and biological processes, and overall soil vitality. In this research in Argentina, the soil labeled before represents a pristine soil with no human intervention. Its chromatography reflects a system in a perfect balance. In the following image, the same soil was covered into an agricultural system. The chromatography result is completely different. The soil is clearly unbalanced. Nutrient availability problems appear not because of pH, but because the biological equilibrium was destroyed. So, what is the alternative? The alternative is agriculture that is as non-invasive and as non-disruptive as possible. What needs to change is the paradigm. In just 1 or 2 centuries, we have pushed this planet to the edge, and we live here. There is no other planet for us, or for our children or grandchildren. Maybe Elon Musk will move to another planet one day. I don't know. The rest of us will stay here. And before we finish, remember 2 words. Repeat them every time you look in the mirror: balance and biodiversity. These words are the key. Don't forget to like this video, subscribe to the channel for more content about agriculture, and share the video. I'll see you in the next episode.